Hi everyone, my name is Daniela Salgado. I'm a first year PhD student at the University of California, San Diego, and La Jolla Institute for Immunology with Dr. Ferhat Ay. And I also did my undergrad here in UNAM. I'm going to talk about a differential method for chromatin contact from HiChIP data. Only a small part from the genome is protein coding, and the other portion corresponds to regulatory sequences in which we have the enhancers. The enhancers can control gene expression, and previous to the capture assays, we were looking for enhancers that were in very close proximity to the gene. But now with the 3D capture assays, we are able to retrieve these kind of examples. For example, here we have a distal enhancer that is controlling the expression of its target gene and even skipping a gene in between. We have this very really nice example from the literature about the sonic heat shock gene that is very important for embryonic development. This gene is controlled by a 1 megabase distal enhancer. And when this is disrupted, we can see an abnormal development both in mouse and human. But how can we capture these chromatin loops? One of the most widely assays used is HiChIP, which mainly relies in DNA cross-linking and then ligation of the interacting regions from the genome and then sequencing. With this, we can extract all possible interactions in the genome, and we end up with a matrix, a contact matrix like this one, where each pixel represents an interaction between one region and region 2. Also, we have this kind of assay that is called HiChIP. Here we retrieve the interactions that are specific to one protein. So we'll have higher resolution regulatory contact maps with much lower sequencing depth. A differential loop can be defined as an interactions with different contact counts between two conditions. For example, here, healthy versus disease. A differential loop can be completely absent in one condition, or it can have differences in strength. Currently, there are some existing differential HiChIP loop colors, but these are not appropriated for HiChIP. Some of the major challenges is that HiChIP is a way more sparse matrix compared to HiChIP, and that we need to address if these changes are due to DNA looping or just by protein distribution. The need of understanding how promoters and enhancers interact together to control gene expression in different contexts, this has led to the generation of hundreds of HiChIP datasets. But currently, there's no proper way to compare differential chromatin loops between two different conditions. The existing differential HiChIP loop methods use RNA count-based methods, for example, DSIC2 and HR, the exact test. Both use a negative binomial distribution. In DSIC2, we use the library size factors and multiply them by the normalized counts. And these normalized counts are modeled by a GLM. Then a gene dispersion is estimated. For the exact test in HR, it's very similar to DSIC2, but here the gene dispersion is estimated in an iterative approach by computing the percentiles and then ranking the genes by expression. HR also implements these generalized linear models, which have an alternative approach for dispersion estimation that assumes the same trend of similar genes with similar expression. Once we assess the dispersion, we want to assess the statistical significance. And we can do this by the likelihood ratio test, LRT, and the quasi-likelihood F test, QLF test. These haven't been explored in HiChIP differential methods. As I mentioned before, the methods that use these differential HiChIP loop colors use RNA-seq count-based methods. And these are not able to model the exponential decay of the contacts with increasing genomic distance that we have for HiChIP data. For this, we propose two solutions. The first one is to weight the p-values by using an independent covariate for each test by the independent hypothesis weighting. Previously in the literature, it has been used the base mean as a covariate for DiffHiChIP, and for HR, we propose using the log count per million as a covariate. We have the second solution where we use the distance stratification. This means applying the differential analysis for individual distance ranges. For example, for the loops that are from 10 kV to 20 kV or 20 kV to 30 kV. And this also has been explored in the literature before. This is our general framework for DiffHiChIP. We implement DSIC2 and HR using the exact test and the generalized linear models with LRT and QLF test. We can take as an input all the loops or the distance stratified loops, and then correct the FDRs using the Benjamin-Hochberg, that is the conventional correction, or the IHW. To validate our method, we use two different data sets. The first one is RNA polymers to HiChIP in colorectal cancer cells. And we call differential loops between two conditions, the control and a CTCF depletion condition. And we extract a lot less interactions in the CTCF depletion compared to the control, as expected. The second data set was a study for skin disorders. Here, they want to model the interferon gamma mediated immune response in keratinocytes. So we call differential loops between unstimulated and stimulated conditions. For this one, we extract very similar counts, very similar amount of interactions between the two conditions and between replicates. So the first question we want to answer is which multiple testing correction to use, the VH or the IHW corrected FDRs. In the HiChIP dataset, we can see that the IHW correction retrieves way more interactions and that the FDR-VH is a subset from this set of interactions. And then we want to address their differential loop sizes. Here in red, I have the union of all the loops in the conditions. So we expect that the differential loops have a very similar trend of distribution of loop sizes. And we can see that the IHW retrieves longer interactions compared to the VH, that is the dashed lines, and that in general, DSIC2 retrieves shorter interactions. Here we show it in blue. Then we want to address how specific are our differential loops to a certain condition. So we perform an aggregate peak analysis. In a contact map, you can see at a loop as a dot. And this highly enriched contact with respect to its background represent the interaction between two regions, region 1 and region 2 in this case. And we can look at other loops in the vicinity and then aggregate their counts. And we'll visualize the counts of a loop and its surrounding. And we can measure this enrichment with an APA score. This is an example. So for example, if you have loops that are gained in a disease, you'll have an enrichment in the background with the disease and not in the healthy. So we expect this pattern for our differential loops. We perform APA analysis in the loops that are lost after CTCF depletion. And we can see that with the IHW correction, we have a more local enrichment. And also we are retrieving more interactions, 400 compared to 20 in the VH model. It's very similar for the GLM models. We are retrieving good enrichment pattern for both VH and IHW, but here the QLF test is the one that retrieves more interactions. And if we look at the differential loops that are only being called by the GLM, also we can see that they are very enriched in the control background. This is an example between MIC and an enhancer. And this differential loop was only called by HR QLF test, not by DSIC or HR. Or HR, the exact method. So here we recommend to use the IHW correction for multiple testing. Then we want to address whether to use DSIC or HR, and if HR, which model. Here I'm showing an example. This is in the HiChIP dataset. This is a differential loop between IER5L and a super enhancer. And this differential loop is only retrieved again by the QLF test and not by the others. If we perform an APA analysis on the loops lost after CTCF depletion, again, we see that the QLF test is the one retrieving more interactions, and even though it's retrieving more, we have a very good enrichment in the control background. And a very interesting result is that when we extract the differential interactions that are only being called by the GLM, these are really high specific to one condition. Finally, as our HiChIP loops should be also called by HiChIP, we address the differential HiChIP loop methods. How can they recover our differential HiChIP? So we saw that the GLM LRT has a better precision, and the QLF tests show a higher recovery of these differential HiChIP loops. Overall, we recommend using the chart GLM with the IHW correction. In conclusion, here I presented a framework of differential HiChIP loop calling. It includes DSIC2 HR using the exact test and the GLM models, and it incorporates the conventional BH-corrected FDRs and the IHW-corrected FDRs. Also, it's important to highlight that our method also supports the utilization of HiChIP data as well. I would like to thank first my supervisor, Dr. Ferhat Ay, and Dr. Sobria, that is a co-author of this paper, and our collaborators and lab members. Thank you, everyone. I'll be happy to answer all your questions.
Thank you for those very useful analysis. Are there questions from the audience?
Hi. I have a kind of like a weird question.
Yes.
When you were talking about like selecting the associations of the HiChIP, but like stratifying within
Yes.
regions, have you ever tried by stratifying not within regions, but like within other HiC profiles that are already produced? So in this way, we can avoid like missing some interactions that might be happening like from faraway regions or within like between chromosomes.
Yeah, so here in this part, we try to use HiC as a ground truth because, yeah, you were saying that the HiC should recover all these HiChIP loops. So this is the only approach we use to compare between HiC and HiChIP. But yeah, it can also be implemented in the stratification to see if, to use it as a helper for defining HiChIP loops. So yeah, we could do that as well.
Thank you.
Yep.
We will move to the next presentation. Thank you, Daniela.
Thank you.